Hello everyone. Today I'm going to discuss a new module called machine learning platform in drug design. So how to create a AI model or a machine learning model and how to use it in your QSAR studies, how to generate an automatic QSAR using a machine learning or AI model, and how to validate the created model using internal validation as well as external validation, how to produce an app, how to share the app across the globe, how to identify the druggable targets, how to differentiate between hits and leads, how to differentiate between a druggable target and a non-druggable target is will be clearly explained today in a brief lecture. So let us go with that app. Now I'm going to I'm going to deal with you an app which clearly says which is designed by an Amrita School of Pharmacy. So this is called Poseidon Q. They also have an what do you say is a global partner, that is University of Digi Study, that is another university from the different country. They are in collaboration. Upon collaboration they have developed an machine learning module which creates the researchers to develop an AI based quantitative structure activity relationship model and helps them to create an automatic QSAR using this method. So in this module, let us try how to do that. Just go for open the software, and this software you can download it from GitHub. It will neither ask you any license or anything. It is an open source software. So just go for data input. Just click here. So now here I'm typing xanthine oxidase. This is the just one of the protein which is sorry enzyme, whichever you say, it's available on the data set. So now upon directly entering here, I have got different types of ChemBL, that is an database where some molecules have been already pre-deposited for this activity. So this might be a different you can use a different targets and different modules and different molecules. So now here in the current study, I am selecting the target one, which is a Homo sapiens, and I am going to extract the molecules from the database ChemBL. And please also note, you can also input your own molecules, which you have already searched through literature and create a different model. And how to use these your own molecules, I'll come up with the next lecture. Let us just go with the current one. And once I click confirm, you can see in the background, it's clearly showing that targets are being extracted. And once it is done, it will show you the overall number of molecules extracted, which will also be presented here on the screen, which will be presented so that you can be able to know so how much molecules have been extracted here. So let us see the molecules already extracting targets 100%. Once it is extracted, let us go for the descriptors. Now, how to identify descriptors? So, what are descriptors? Descriptors are those molecules which, sorry, those attributes which you assign to your model. That means in any model, if you create, you have to cluster your data. What do you mean by clustering? That is segregation of molecules based upon their attributes. These attributes may be physical, chemical, biological, or any other. Sometimes these attributes may be substructure. Suppose if you have a big structure, you have 3 or 4 small molecular structures. You can also call them as substructures, also called as chemical fingerprints. Sometimes you have a descriptors. The descriptor may be number of hydrogen bonds, number of hydrogen bond acceptors. It may be polar surface area. It may be any other modules. which you can execute the data set. So this is the fundamental information which you are going to get through the drug design module. So further information, let us see what exactly it happens on the data set and how it gets confirmed. So here, so let us change the data set. So here it is not extracted. Let us change the data set to another data set. There may be some issue with the data set which is selected. So now I'm going here. I'm selecting the target 10. So now I click extract. Now it say take extracting data. I'll click OK. Now you can see in the background, it is showing that extracting target molecules with IC50 values from the ChemBL dataset. So now see, it has extracted at least 56 molecules. I'm just clicking OK. After that, till now I have discussed with you regarding the modules of how to use the descriptors. So here now I have selected RDKit descriptors. What do you mean by RDKit descriptors? Are attributes which I am giving to my chemical structure. These attributes are hydrogen bonds, PSA, as I've discussed. Let me show you. Once I click on confirm, you can see in the background, it is generating a bonding box. What do you mean by bonding box? It uses various physicochemical descriptors as a value of IC50 value. That means one ordinate with which you are analyzing the IC50 value will be your physicochemical descriptor, and the other ordinate, that means which will be plotted against, or that will be the IC50 value. That means if a molecule has a molecular fingerprints with number of hydrogen bonds, number of acceptors, number of polar surface area, and log P value, TPSA values. There are several attributes which you can see on my left side of the screen, with which you are correlating your IC50 value. Now I'll just click OK. Now I'll go back to the software. You can see these many attributes have been assigned to the IC50 value of to the total number of 56 molecular attributes. Now, first thing, how you will know whether your designed or created module is correct or not. That means again that the basic statistics and research methodology, which says you that your attribute should have a best line of best fit. It should follow the straight line. We should have R square near to be 1, and there are several more factors. So here I am selecting histone gradient. Now first, before going to any model, I'll just click on compare. While comparing, now this is a k-fold split. What do you mean by k-fold split? Here in the k-fold split, it is splitting the overall data set into different modules or different clusters, based on which it will cluster them based upon highly active molecules, moderately active, lowest active, and inactive molecule, based upon all these physicochemical attributes. Now let me click onto the RDK descriptors. Now let me click confirm. Once I do that, you can see there are several modules or models are being running in the background, which clearly says that, you see, this is 0.48, 0.34. Now this is a very bad model which should not go, and the model is not near to the R-square value. So this is the wrong model which you should not accept. So now it asks me about tanimeter report, and it gives me RMSE value and everything. So always you should be very clever enough in identifying your data set and doing something. So let me go with another input. Let me check with a different data set. So let me change with another module. So now here, now I'm selecting a different data set. Now I have gone with DNA topoisomerase. Now I am clicking extract. Now again it's showing me. So earlier in my previous search or screening module, where I am able to screen out only 40 to 50 molecules, here you can see the data set is increased to 337. What does it mean? As the data set increases, the statistic number of sample size increases, you'll get the better results. So that is what is being analyzed. This, and for this you require a basic understanding about statistics and research methods. Let us go with the descriptor count, and now we will go with RDK descriptors. Again, I already explained what are RDK descriptors. Further, without wasting any time, I'm going to get confirm modules. So now you see, again, it will be designing the binding box. So now I just click OK. Now it will analyze the modalities and gives you the best binding results out of the models. Now you see the binding box is very click. Now let me compare this binding box. Again, it is splitting the data set. Let me check with just click confirm. Now you'll get different modules. So you should have an machine learning model with better R square value with which you can correlate your data, and further you can have an best validation, that without any validation any data is flu or flawless. So in order to do that, your data set should be, see, your data set is not that good, but it's not even bad. Let's go with this data. So let us create an folder for this. Now in this, I'll create a new folder. I'll just create a name, that is YTB. So, and just select the folder. Now the mean and SD values are already saved. I'll click OK. I'll close this window. Now here, I'll click the stone gradient. Again, I will click on to the RD descriptors. Now I'll put confirm. Once I do that, my model is running in the background. Now I'll get the best fit of all the attributes, and it will be tuned with this automatic tuning method. Just you click on that. Now your machine learning model will be built in the background. You need not do anything on that. This software is so user friendly. It will create your own model. So now again, I'll go back to my app, which I have, sorry, on the working directory, and I'll just click save. It's okay. Now you see this value, that is RMSCV 0.554. However, this is not that perfect. And see, this data is already clustered, and you should select the data only which has an R square value nearer to the value of 1. That is, it should not be 0.5. And this is a poor data. However, let us go and see how to use an app. Then I'll click produce app. Just I will give the app title DRV doc, and I'll just click build. Once I do that, again it will ask me the working directory, that is YTB, and I'll just click select folder. And now it says that your app is developed, and go and build your web app. Now I'm just closing it. Let us see what exactly my working directory contains. So this working directory, if I open it, it has a scatter plot which clearly differentiates the overall data, and see the data sets. So this is one of the data, and you can see the Tanimeter report, that is similarity index, and also it has an outliners. Also you can see the total train set and test set. That means overall the 340 plus molecules, it is divided into train and test. You can open it and explore what are all the chemical descriptors, that is RD kit descriptors used in creation of model, is already mentioned here on the header. And you can just close these and you go for the app. You can see the various data available here, and you can just click. This is a binding efficiency based upon your data set, and this is the what do you say, the neural network, and as well as local decision tree, how the decision is made for your small molecules, on what basis of the decision, how your data set is differentiated. It is already given with this what do you say, decision tree analysis. Further you can see further more data, and you can see this is your decision tree, and this is your scatter plot, this is your Tanimata report, that is similarity index. This is all what, and this is your computational chemistry by using Python language, machine learning. It is creating an app. Now the first more thing is, you have been internally validated segregation of data into training and test, and how you will analyze and how you provide a web app. That is a big task in this software, which requires some knowledge, basic knowledge. Let us try that. For that, you have to go for GitHub, create your account using a simple Gmail account. You need not go for the free account. This is my GitHub account. I'll just click create new repository. So you should also have a GitHub account. Get your GitHub account. Now I will give the name YTB. YTB code is available. Always keep your GitHub repository as public, and just click create repository. Once you create repository, you got an link for your GitHub. Actually, what we are doing here is, the data which is available on my desktop, I'm transferring it onto the GitHub folder, where I can create a repository for my molecules or the data set, and using that repositories I can create an app. So first I have to transfer my data set onto the GitHub. How to do that, I'll show you. For that, you need to download Git CMD. So how to download, just go for Git CMD download. I'm just showing you step-by-step process. So here, download Git, you go for this Git download. Once you download, it will come in this fashion, Git CMD. I'm just showing you. Here I have to run some commands so that I will make my working directory, which is already on my desktop, to go to GitHub. For that, I have to go to my command prompts. I have to click my desktop. Then again, I'll use my folder, that is YTB. Now my folder is ready, and I'll go for app. My app is ready. Now I'll go for Git, and I'll click initiate. And also once I click enter, now in the GitHub is already initiated on my working directory. Now I have to give certain commands so that my working directory is completely accessed onto the GitHub folder. How to do that? Just click on Git space. Now I have a commands. I will show what type of commands are there and how to use it. These commands will be made available to everyone. Now here, next one is Git. Go for remote, add origin. Now I'll place what is happening here is the Git, and I'm making a remote directory, that is which is on my desktop. I'm adding it to the origin, which is on the GitHub. This is what is the technical language of which I'm transferring my files from my desktop to GitHub. I'll just click enter. Now it says it's executed properly. Now I'll click Git space add and space dot. I'll click enter. Now this is what the molecular fingerprints are being made and which has been transferred onto my working directory. Further, I'll go for Git commit -m. So let me check the command. Git commit OK -m. So here what it is showing is that this command, if commit do something if the command is right. That is what is the coder has set some commands for the software. I'll just click enter. Now you see all the tomato plots, fingerprints, everything has been executed because every command was perfect. Now let us go with another command, that is again go for git space branch minus, again I have to click m space main. I'll click enter. Now my branches are made. Now I have to push. See, the word pull and push commands are that which enables to transfer my files from my working directory to the GitHub repository. So this is what you should understand why I am using the repository. Repository is a place where I am storing my data. So GitHub is my repository. I am working on my desktop. I'm transferring my data from my desktop to the GitHub. This is what is called repository analysis. Okay. Now I'll give the final command, push -u origin space main. Once I have executed this, you will get a complete report of your data. This is enumerations, countings, compressions, transferring to the GitHub. Everything is done. It clearly says that everything of your data is being transferred from my desktop to the GitHub. Let me check whether it is correct or not. I have gone to my repository. My repository name is YTB. I am clicking on that. See, all my computational chemistry, all my app which I have generated, all my TXT files, Tanimeter reports, everything is transferred onto the GitHub. Now, see, I'm using GitHub as a repository only. I'm not producing any app out of it, nor I'm analyzing the app which I have produced. For that, I have to use another module, that is Streamlit. So now this Streamlit, I will click free version on this. You can also try that. You have to use your login credentials, which integrates your GitHub with your Streamlit. Now here you have an option called create app. Here it would ask you about deploy now. You can click here and select that module. This is my app. Now I'll just go on to prediction. I'll give some name, that is YTB1T8i. This is my code. And now just I'll click deploy. What happens is that all my data which is available in the repository will be transferred onto the GitHub, from the GitHub to the Streamlit, and I can generate my app. So this app is taking some time to develop. So without wasting any time, let us check already developed apps which are available on my, my Streamlit directory. I'll just show you how to use them. So these are all the apps. So these are used with different data sets. So let me go with this. This is which I was doing it right now. So I should not have checked out this. So I'll go with this module. So this is my app. Let us see what it shows. Okay, there is some again. Let me check with this one. So okay, let me check with this. Okay, so in this format, your module will be ready. How to check? Let me check this. I'll click confirm. Once I do that, I will get the data. this data itself shows that is out applicability domain. What does it mean? I have generated a domain or knowledge, or that is called clustering or AI module, which clearly says that my data set should be of certain parameters, which will give me. See, again, this is an out applicability domain. It is out. So let me check with some data sets which I have with me from other, what you say, other molecules. Uh, let me check from this. So this is my personal data, which is not, published or anywhere. So what I'll do, I'll just click on this data set and I will check whether my data set will be useful in doing something. I'll just click confirm. You see, this data set is within the applicability domain. That means the domain which I am searching for, that model which I have done, if I am using this molecule will be perfect for prediction, and you will get the PIC50 value also. That means predicted IC50 value. However, my Streamlight is overloaded with my repositories. That's why it is unable to show. Let me check with the current repository whether it has been done or not. So it depends upon how long you are using your. Let me delete much of the repositories so that you can freely check the data. So all the data which are not useful or not used or been thrown off. Now let me check what happens with this data set, whether it will show you my developed app. Okay, which is able to communicate something because I have cleaned my all my apps which are not used. So what happens with the molecular fingerprints with an developed model? It will check the availability of the molecules which are designed on computers, whether they will be good enough for execution in the labs, which will be predicted using the AI module. This is the basic fundamental, and you should always know that the in applicability domain value should be more than 6 for the optimal fit of the model, and your R square value should also be highly tunable. So I'm sorry, so that I'm unable to show you my data set because of overload on my data. Let me try with something else. It might be because of speed of my computer. It might be because of I'm opening the multiple modules. There are many more things which I cannot, which I'm not interested to comment. See, this is again. So you will have a PIC50 value structure of your, what do you say, the will be clearly loaded onto it. Let me again try back using the Streamlight. Last try. You can do on your own later on. Okay. So because again, just try the PIC50 values, structure values, everything will be clearly mentioned. Why these many iterations? Because of the space, a lot many things. So I'm stopping here by giving you the best modules of, and you can directly share this app to anyone. You can write an email. Suppose I'm just typing my own email ID here. How to share an app, gmail.com. I'll just invite. What happens? This one, or you can write any other email. So let me check with. So just you can invite. Okay, you can even copy link and you can paste anywhere. You can do it on social LinkedIn. You can do it on any other forum, and you can even so that someone can explore. So now I have copied the link. I'll just paste. I'll click and I'll close all these things. Let me check what exactly my Streamlight shows. So this is again because there is some technical error with the Streamlight, which I am not going to solve. So I'm stopping here. I'll hope you can create your own models and design, and you can identify whether which is within the applicability domain or not. Thank you.